The moment the heart stops, the body does not stop. It begins something else entirely. Within 4 minutes of death, cells that have been running continuously for decades suddenly lose their power supply. They do not simply shut down, they begin to consume themselves. The process is called autolysis. It is the body's own enzymes, released from their usual containment, beginning to digest the surrounding tissue from the inside out. The body, in its first act after death, starts eating itself. This is not decay in the way most people imagine it. It is not an external process. It is the machinery of life turning against the structure that housed it, the moment that structure stops providing the conditions required to keep the machinery in check. What follows is not chaos. It is a sequence, a predictable, reproducible, scientifically documented sequence that follows the same basic arc in every human body on Earth. That sequence has a name. It is called decomposition, and the science that studies it is called taphonomy, from the Greek taphos, meaning burial, and nomos, meaning law. Taphonomy emerged as a formal discipline in the 20th century, largely through the work of researchers who recognized that understanding how bodies change after death was essential to understanding everything from the fossil record to criminal investigations. The person most responsible for turning this into a modern science was Dr. William Bass, a forensic anthropologist at the University of Tennessee. In 1981, Bass established what became known as the Body Farm, a research facility where donated human bodies were studied in open-air conditions to document exactly how decomposition unfolds over time. The Body Farm produced data that had never existed before. For the first time, forensic scientists had a reliable, peer-reviewed timeline for decomposition under varying conditions of temperature, humidity, insect activity, and exposure. What that data revealed was not what most people expect. The body after death is not passive. It is extraordinarily active. Let's walk through what actually happens. In the first few hours, 3 simultaneous processes begin. Pallor mortis. The skin loses color as blood stops circulating. The pink flush of living tissue drains away within 15 to 25 minutes of death. Livor mortis. Blood, no longer pumped, begins to pool under gravity in the lowest parts of the body. The pooled blood darkens the skin where it settles, creating distinctive discoloration. This begins within 2 hours and becomes fixed in place, meaning it stops shifting within 8 to 12 hours. Forensic examiners use the pattern of livor mortis to determine whether a body was moved after death. Algor mortis. The body cools. Living bodies maintain temperature through metabolic heat. Without that heat source, the body temperature drops at a predictable rate, roughly 1 to 1.5 degrees Celsius per hour under standard conditions, until it reaches the temperature of the surrounding environment. These 3 processes together, Pallor, livor, and algor mortis, give forensic scientists their initial timeline window. Then comes rigor mortis. Between 2 and 6 hours after death, muscles begin to stiffen. Rigor mortis is caused by a chemical change in muscle fibers. Without ATP, the energy molecule, muscle proteins lock into a contracted state and cannot release. The stiffening moves from the face downward through the body and becomes complete within 12 hours. It then holds for another 12 to 24 hours before beginning to release as the muscle tissue itself begins to break down. The progression, the hold, and the release of rigor mortis give investigators another precise timing tool. While all of this is happening on the surface, the inside of the body is undergoing its own transformation. The human digestive system contains approximately 38 trillion bacteria. In life, these bacteria are carefully contained within the gut, separated from the rest of the body by barriers that require active biological maintenance to hold. When those barriers fail, the bacteria do not wait. They begin migrating through tissue, through blood vessels, through the entire internal structure of the body. They produce gases as they metabolize: hydrogen sulfide, methane, ammonia, carbon dioxide. These gases build pressure inside the body, causing the bloat stage that researchers identify as the second major phase of decomposition. The bloat stage is also when the body first becomes visible to insects from a significant distance. The gases released carry volatile chemical compounds that certain flies can detect from a kilometer or more away. Blowflies are typically the first to arrive, often within minutes of death in warm conditions. They lay eggs in natural openings of the body. The eggs hatch into larvae. the larvae feed. This is not accidental. It is an ecological relationship. Insects, bacteria, and the body form a system, a decomposition ecosystem, and each component of that system is responding to signals produced by the other components. Dr. Jason Byrd, a forensic entomologist, has documented how precisely insect arrival and development patterns can be used to establish time of death. Different species arrive at predictable intervals. Their developmental stages follow temperature-dependent timelines. A skilled examiner can work backward from the insects present to determine when death occurred, sometimes to within hours. Following the bloat stage comes active decay, the phase in which the largest volume of body mass is lost in the shortest period of time. Internal gases escape, tissue liquefies. The body loses the majority of its mass during this phase, which can last from days to weeks depending on environmental conditions. What the body leaves behind during active decay is called a cadaver decomposition island, a term coined by researchers studying the ecological impact of a decomposing body on the surrounding environment. The fluids released during decomposition are extraordinarily rich in nutrients: nitrogen, carbon, phosphorus, sulfur. They saturate the soil in a concentrated area around the body. That saturation kills some plant life initially. Then, weeks or months later, it triggers explosive growth. Plants in the zone around a former body are detectably different from surrounding plants for years afterward. Soil chemistry, microbial populations, insect communities, all are transformed by the passage of a body through that specific patch of earth. The body does not simply disappear into the ground. It redesigns the ecosystem around it. In the final phases, advanced decay and skeletonization, the remaining soft tissue is consumed, and what is left is bone. Bone itself is not inert. It continues to change. Lipids leach out of bone over time. Collagen degrades. Mineral structure shifts. In the right conditions, bone can persist for thousands or even millions of years. It can carry the chemistry of a person's diet, their geographic origins, their diseases, and their age at death, all readable by scientists long after everything else is gone. This is what taphonomy offers, not just a timeline of dissolution, but a record. Every body leaves a signature in the environment it passes through. The bacteria, the insects, the soil, the nearby plants, all carry information about what was there and when and how it changed. Death in this framework is not an ending, it is a transfer. The atoms that built a human body, the carbon, the nitrogen, the calcium, the iron, do not leave the biosphere. They enter a new cycle. They feed bacteria, which feed insects, which feed birds. They enrich soil, which feeds plants, which feed animals. The body that stops is the beginning of something that does not stop. And every step of that process, from the first act of autolysis in the minutes after the heart stops to the chemistry of bone 1,000 years later, follows rules that science is still in the process of fully mapping. The body after death is not still. It is just moving in a direction you don't usually watch.